Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Addressing Pre-Analytical and Processing Variables in Plasma Samples Destined for Multi-omic Analysis via an Operated Independent Plasma Quality Control Measurement. My name's Charlie Carter, and I'll be moderating today's presentation. I'm delighted to be joined by Dr. Andy Brooks, Chief Operating Officer and Director of Technology for Rutgers University, and Dr. Kevin Lance, Marketing Manager at Unchained Labs. Today we'll be exploring the development and implementation of a novel plasma quality control approach on the Lunatic microvolume UV vis spectrometer from Unchained Labs. Please feel free to ask your questions for the Q&A session at any time during the webinar. You can submit your question at the left of your screen. So without further delay, I'd like to hand over to Andy and Kevin.
Thank you for that introduction, and thanks to everyone joining in on this webinar. Now I'll introduce you to some of the instrumentation being discussed in today's webinar, the Big Lunatic. In a lot of things in life, it's important to check for impurities before going on to the next step, to make sure the things that shouldn't be there aren't. Like this bowl of cereal, you really shouldn't take a bite before you've taken a look. In this talk, we'll be looking at a technique to help you check your plasma samples for impurities before going to the next step. Big Lunatic delivers full spectrum UV vis absorbance for sample quantification from only 2 microliters of sample. Big Lunatic is a plate-based system for high throughput or automated quantification of up to 96 samples in about 10 minutes. One secret to Big Lunatic's success is its consumable plate. Each well of the 96 wells is a microfluidic circuit that detects absorbance from 230 to 750 nanometers along a fixed path length through a microcuvette. Fixed path lengths make for accurate and precise concentration measurements, since we are always sure of the path length to be used in Beer's law calculations, and that path length is not moving around during reads. Fixed path lengths mean you know exactly what you're measuring. For the plate shown here with 2 microcuvettes, absorbance is covered over a wide dynamic range of 0.03 to 275 OD. The unique microfluidic circuits molded into Lunatic plates and chips ensure every 2 microliter sample gets its own space, and there's no cross-contamination between samples run sequentially on the same UV vis pedestal or probe. How does Big Lunatic handle a read? Well, samples are deposited into this input well and drawn into the storage channel by capillary action. A sample can be left in the storage channel for up to 2 hours without worries about evaporation. And then during a read, Lunatic uses a pump to aspirate that sample further into the microcuvettes. Shown here are high Lunatic strips with 2 microcuvettes in each circuit, with a 0.1 and 0.7 millimeter path length. The 2 different size microcuvettes make the wide dynamic range possible, and the 0.1 millimeter cuvette provides a short enough path length to enable a high maximum detectable absorbance. Because Big Lunatic detects with great precision and accuracy across the full spectrum range, that means you can get quantification any way you want on protein samples, nucleic acid samples, or, like we'll see today, even complex samples like plasma. The software also makes it easy to analyze things just the way you want it, with different ways to correct for sample background, or the option to deconvolute an absorbance spectrum to help identify impurities. It will even make it easy to run colorimetric assays like Lowry, Bradford, BCA, or Psi3 and Psi5. When we're talking about UV vis absorbance, here's a quick example of using Big Lunatic to quantify a protein sample. We know proteins peak at 280 nanometers, so that's the wavelength our A280 applications focus in on. We can see in this screenshot that the application is determining an absorbance of just over 4. To get to concentration, we use the E1% extinction coefficient, which is necessary to determine concentration from light absorbance. The E1% can be determined by measuring the A280 of a 1% solution of a protein, or it can be calculated based on protein sequence. Combining the absorbance of just over 4 with the E1% of 10 is what gets us to our A280 concentration of 4.22 milligrams per milliliter. This application also reports the background contribution due to turbidity and the A260-A280 ratio as quality metrics typical for protein quantification. So that's a brief primer on using UV-Vis for quantifying concentration using a simple protein as an example. Here we're looking at a huge protein dilution series of IgG and PBS to show off the linearity of Big Lunatic and how Big Lunatic is the best system out there to measure biologics at high throughput and high concentration. On the left we can see the absorbance of those samples, and on the right we can see that after quantification their linearity gives an R-squared value of 0.9999. Big Lunatic has a dynamic range that covers from 0.02 mg per mil to 200 mg per mil for an antibody, so users can run any protein without ever having to dilute again. And precision and accuracy is spot on, with results within 2% for accuracy and 1% for precision when checked out with analytical standards. When it comes to plasma quality assessment, plasma samples can have the same visual appearance and color and clarity, but have completely different biological components. Depending on concentration, two different mixes of hemoglobin and bilirubin can look the same. Big Lunatic can analyze the total absorbance spectrum to isolate and quantify plasma sample turbidity, bilirubin, hemoglobin, and protein. In the example shown here, Big Lunatic analyzes the peaks in the 350 to 550 nanometer range to determine the sample contains 400 milligrams per deciliter hemoglobin and 1 milligram per deciliter bilirubin. This is an approach to score plasma quality more objectively than the previously used visual assessments. And Big Lunatic's changed up a bit over the years too. It used to formerly be called the Trinian DropSense 96, and that's now called the Big Lunatic. And the software used to be called either the C-Drop or Drop Quant Data Viewer, and now we'll call it the Lunatic and Stunner software. And Big Lunatic also talks to robots. You can hook it up with any liquid handler system and run your measurements completely hands-off with our API setup and spend more of your precious lab time somewhere else in your workflow. So with normal UV-Vis, it can be hard to spot what you're looking for when you're looking for things that are hidden in your sample. But getting more with accurate UV-Vis quantification, then you can zoom in and reveal exactly what's there. And with that, I'll hand the presentation off to Dr. Brooks for the next section.
Thank you for having me. It's my pleasure to talk with you today about the integration of technology, process, and quality control that's used in biobank settings and a number of genomics and other omics-related applications in order to protect the complete physical and digital sample chain of custody. A lot of different things are done with biomaterials, and what we do today may not very well reflect what we do in the future. And we have to think about the one thing that will be constant for the current technologies that are being used today and those technologies that will be used in the future, and that is the biological sample. In today's day and age, we use biological samples for research and development, biomarker discovery, for targeted clinical trials, rational drug development, the creation of new companion and molecular diagnostics. And really, if you use DNA as an example, this all was accomplished in a very short period of time. It began in the 50s with Watson and Crick's discovery of the structure of the DNA double helix. And in the more recent future, with the sequencing of the human genome, we've applied a lot of data to the development in these areas. And the future of the use of biological samples is even more promising. In the very near future, we'll have complete integrative wellness monitoring, looking at our entire picture of health and disease. We can do complete health prediction based on genetics and environment, be able to do genome sequencing as early as 6 weeks in pregnancy beyond trisomy and other disorders that are measured now. We can develop a comprehensive molecular monitoring program for all diseases and therapies. And this will transition even to point of care in looking at testing and rapid testing in doctors' offices, clinics, and hospitals. And again, the one thing that's consistent with these themes is the quality of the biological sample collected, how it's processed, and how it's assessed for quality control in order to be able to make these types of measurements. It's really important to understand the difference also in collecting biomaterials and biological samples. What's the difference between a stored sample and a biobank sample? A bank sample is one that's proactively acquired for future testing or analysis. So not necessarily a question that we're going to ask now, but making sure that when we bank a sample, we understand everything about the metadata and the quality of that sample, because it may be a long period of time before it's tested. And a bank sample is also sent to multiple different recipients. So the way in which we collect and process and store those samples to prevent freeze-thaw, to prevent changes in the analytical or functional quality control is also really critical. And lastly, a bank sample should never be depleted. So doing upfront and integrated quality control is important to prevent the unnecessary freeze-thawing of samples to make assessments later on. Controlling pre-analytical variables is part of an integrated process. And when we're going to look at the quality control of biomaterials, it's a function of this exact process, as we're going to share some data with you during this presentation. So let's just assume for a second everyone coordinates around a single technology for nucleic acid extraction. Above the dotted line is what a typical lab may do. There's manual processing. The vials or the storage vessels that are chosen may not be automation friendly. A lot of sample is used to make some blunt measurements. We make eyeball measurements when it comes to volume. And then for QC, we use rather blunt tools for looking at whether or not nucleic acid is of quality enough for analysis. Below the dotted line is really the approach that we've been using and that other labs are incorporating on a more routine basis. And right in the middle of that process, you can see the Unchained Lunatic, which is a critical component for Analytical QC, even when we're talking about DNA extraction, but plasma and other molecular components. So below that dotted line, there's automated processing. We use tubes that are designed that are automation friendly to facilitate cost-effective and quick processing, but also give us complete chain of custody for the sample. We use technologies like the Unchained Lunatic for cuvetless spectroscopy, not only to get very accurate measures and quantitation for nucleic acid, but also to look at contaminant profiles that could lead to technological variation in a sample that could be interpreted as biological variation. And that's the real important component about quality control. Downstream of that, we use ultrasound detection measurements for looking at volumes. And then functional QC, which is not the focus of the presentation today, but is also an important component to make sure that we have the right sample and that that sample is qualified for downstream analysis, whether it be genotyping, sequencing, or other applications. And the way in which we do research and the standard of care is changing, and now everyone's interested in multi-omic variation, and rightfully so. And if you look at a function of the genome, epigenetics, gene expression profiling, protein analysis, and metabolite profiling, it's the combination of these different omic variations in coordination with environmental exposure for gene-environment interaction, which determines health or disease. But on the left side of this slide is what's really important. If we look at the scope and scale of how we collect and analyze or quality control biomaterials, we need to have technologies that do it cost effectively and efficiently, because the numbers of samples we're talking about has dramatically changed over time. And if we apply these principles on the right hand of the slide, it's only going to continue to grow. So imagine with the almost 350 million people in the US in 2019, there were 125 million hospital visits, almost 1 billion doctor's office visits, and there were 13 billion diagnostic tests performed, with less than 10% being molecular analyses. Now imagine if we only collected 5 biosample aliquots for each person, a little bit of urine, a little bit of plasma, maybe a little bit of serum, some DNA, and some whole blood for future use. And it's very easy to get well past 5 different biomaterials. That would be 4.4 billion samples a year that we would have to pre-process and quality control. And imagine if we collected 5 biomaterials each year for an average lifespan, in this instance of 50 years, that's 220 billion samples that we would have under management, quality control process, or what have you. So it's clear that molecular samples will far exceed the number of paraffin blocks that are used now for tissue-based applications or pathological evaluations by some orders. One thing that's consistent and continues to be consistent and a driving factor is science and technology leads innovation. On the right-hand side is a molecular cartoon, if you will, about the different applications from the four pillars of biomaterials that exist here. And it's really critical that we integrate sample collection, the processing, and quality control with an eye on all of these applications. We also need to understand the power of evolving technologies and develop a sample-centric roadmap for future use. In order to develop anything that's sample-centric, we need to make sure we lead with quality control assessment. So by creating quality control metrics that can be standardized across all collections, we can compare samples across biobanks, across clinics, across programs. The creation of this resource will allow us to seamlessly integrate both academic and industry applications if we can all agree on sample quality control measures. So when we think about quality control, we start with the sample, and there are three real areas that you could have adverse sample quality events. The sample could be collected incorrectly, the sample could be extracted or pre-processed incorrectly, or the sample could be stored incorrectly. Or usually it's not incorrectly, but it's a variation of these different activities that leads to variability that could be misinterpreted as biological variability, which is what we're trying to get at, to understand different biological perturbations, different disease states. So in order to be able to control, if you will, for all three of these variables, we need to be able to look at measures in the sample and then be able to understand where that impact was. So at the time of collection, if the sample takes too long to be processed for blood, we might use hemolysis as a measure in plasma of poor collection. If a sample gives low yield or actually has a contaminant profile that's a function of how you extracted the sample, then we could look at extraction being a variable that prevents the performance of the sample. And over time, if storage is not done correctly, we could see changes in cellular programs if they require preserved cells. We can see degradation in nucleic acid. And all of these things are tied to quality control. So getting the measurement right at the time the sample is collected and processed is critical for understanding all of these parameters. And what we've done is we've incorporated Unchained Lunatic into this process for a cost-effective and efficient and high-quality assessment of sample quality. The first QC approach I'd like to talk about is plasma QC. The biological relevance of plasma QC until recently is really largely unknown. The color and clarity gradings that are done, whether we look at whether a sample is hemolyzed, which would lead to a pink-red color, if the sample is icteric, which has an abundance of bilirubin, it could be orange or dark yellow color, and then the turbidity of the sample, whether it's transparent versus opaque, all have different impacts on downstream analyses. And until recently, most labs, and still a lot of labs, will use a visual grading score for looking at plasma quality, either 0 to 5, maybe 0 to 10. And the fact of the matter is, the ends of the spectrum are usually accurately recorded, but it's very hard to get a technician, no matter how they're trained, and even if you look in the middle image here for icteric serum, to get a difference of opinion without a visual standard curve for samples. So using the Unchained Lunatic, we were able to work with Unchained to actually quantify the contaminants that are a part of any potential plasma or serum sample, looking at hemoglobin, bilirubin, and turbidity as a function of downstream performance. It's a validated reference analysis that provides a detailed QC using raw UV vis spectrum and unmixed spectral filtering. So what we can actually get is a quantitative readout of hemolysis, of bilirubin, how icteric the sample is, and how turbid the sample is, which would depend on any kind of fatty deposits or things that might be in the sample to affect performance. Now, having this data doesn't necessarily give you an answer. We're going to show some correlative data for downstream analysis in a moment. But what it does is it allows you to qualify each sample. So for instance, if we're doing a study where it takes time to get sample back to the lab, or it has to be shipped overnight, and all samples are processed within, let's say, 24 hours of collection, it allows us to assess the relative quality of each sample when they're being chosen later on for downstream analysis. And this kind of assessment is more subjective versus objective scoring of quality. So here you can see, if we were to look at the intersection of bilirubin on one axis and hemoglobin on the other, even though a technician, when they're doing their individual scoring, can determine where you are on a continuum of, in the case of bilirubin, the richness of a color, the darkness of a color. For hemoglobin, whether there is a color versus, or no red color versus a pinkish or red hue, and then turbidity. But what we can't do in our individual scores is actually look at all of the components and then come up with an assessment of overall sample quality. This approach, and having a quantitative measure to give us a true set of scoring quality metrics, is much more efficient, much more reproducible, and has not led to really subjective decisions by a tech in their assessment. And it's not very different than how you might assess something like contact inhibition. So if we look at confluency for cells, let's say, depending on where you look in the dish and the technician's mechanism for searching quadrants in the dish, you could get very different assessments of confluency. Same thing for this. By a given tech on a different day, you might get a different assessment of quality if you're matching it up against a visual standard. So what does this mean? Here are two examples, actually, of how plasma QC can affect some downstream applications when you're dealing with plasma. And we do a lot of work in cell-free nucleic acid, and it's really critical that we get the highest quality cell-free nucleic acid from every plasma sample for a number of reasons, largely given that we want to have the most complex sample. That if the sample is degraded or further degraded, or if the yield of cell-free DNA is impacted, that ultimately can affect the complexity of the overall sample when we're looking at an entire free-floating genome, or a cancer genome, or a tumor genome, if you will, from free-floating DNA. So if you look on the left side, the yellow transparent sample has an average value of about hemoglobin of 10 mg per deciliter. This is yellow and golden colored, often to be mistaken with an apple juice kind of color, with a bilirubin and turbidity with measurements of 0.4 mg per deciliter and a 0.10 D660. But if we look at a dark yellow sample that's still transparent, so it's not turbid, you can see the big difference in bilirubin to 3 mg per deciliter. And it's very easy from across the room here to be able to determine the difference in the spectral analysis for the curve, which gives us accurate quantitation. But the most important piece of information on this slide here on the bottom is that we saw in this sample, or in these samples with this profile, close to a 25% decrease in cell-free DNA and an 18% decrease in sample performance, meaning that the complexity of that sample due to a decrease in DNA led to different results, something that we would have interpreted different biologically. And if we look at hemoglobin contamination as an example, very similar. So here's the standard, the gold standard, if you will, or the yellow standard of the very clear, transparent, golden yellow plasma sample and the spectral profile for that reference sample. And then a pink sample, something that's pinkish, not dark red. We do see samples that look almost like blood following separation because the hemolysis is so great, but this is just a pink sample, if you will. And here you can see a difference to 140 mg per deciliter. So just even the most pink sample has a huge increase, or a large increase in hemoglobin. And in this instance, we saw a 31% decrease in cell-free DNA yield from the same sample that had that kind of quality where the sample was not hemolyzed, and a 26% decrease in sample performance. And these are significant when you're talking about analytics that require a high level of sensitivity. So spectroscopy analysis for plasma QC accomplishes a lot of different things. It enables a high throughput analysis of plasma samples. It's operator independent. So the objective scoring of 3 biologically relevant plasma parameters, hemoglobin, bilirubin, and turbidity, have a big impact on performance and sample quality, and now doesn't require the operator, like the image below, to be able to choose a score based on these different shades of gray, if you will, or shades of red or shades of yellow. It allows us to qualify samples for downstream performance using the data-driven binning of these quantitative cutoffs. Again, every application may have a different impact based on the quality score. So it's our job as scientists to use that data to better identify samples and or create cutoffs for our downstream analysis. And the amazing thing is the cost is pennies per read, and the complete analysis uses about 2 microliters of each plasma sample. So there's absolutely no waste of primary sample, and this allows you to do this correlation to quality prior to any downstream analysis. So it's not a matter of doing a huge sequencing experiments only to find out that the complexity or that the quality of your cell-free DNA was a function of plasma QC. DNA QC is another component that we use the Lunatic for in our process that's also critical. And we break up nucleic acid quality control into 2 compartments, if you will. Analytical QC, where traditionally this is the static biological storage QC component, where we can measure sample concentration, the purity of the sample, yield, and volume in a non-contact measurement. And then there's functional QC. So this is the dynamic discovery component that is not incorporated by a lot of labs. It is by a lot of biobanks, but not necessarily a lot of individual labs. And this component looks at contamination, the performance of the sample, or being able to predict the performance of the sample, the fingerprint to know that the sample is unique, and we can also incorporate the screening of subjects for clinical trials. There have been a lot of advances in analytical QC, largely with the Unchained Labs Lunatic, because this cuvetless spectroscopy, unlike a nanodrop or some other measures, actually have the ability on the right-hand side of this slide to look at and determine via content analysis any potential contaminants. A very similar approach to what I just mentioned about for plasma or serum QC. We can look at contaminants in the sample, but not biological contaminants per se, because if we're looking at nucleic acid, and the nucleic acids have been purified, if you will, or enriched, we're not going to see hemoglobin or other things. The ratios will tell us if there's protein contamination. But the biggest impact is looking at what other things are in that sample that could lead to a unrealistic or faulty quantification or inhibition of downstream applications. So if you look at this slide, you can see on the left is a reference list of measurements looking at what double-stranded DNA, pure double-stranded DNA, RNA, protein, or phenol looks like. And on the right-hand side is the actual measurement of a sample. So what we can do is look at the deconvolution of absorbance profiles to determine, based on the reference spectrum, what the contaminant profile is. There's a large dynamic range for analysis here, and the data can be analyzed repeatedly. But what it allows us to do is focus in on exactly what the concentration or amount of each contaminant is, and with our functional studies, determine the impact of that contaminant on downstream applications. Using the Lunatic with the C-drop analysis, if you will, it gives a reading that is almost superimposable on pico green, which we know is accurate because the dye only intercalates double-stranded DNA. And if you can see this as a function of different quantities of contamination and different contaminants, that the Unchained Lunatic calculation and being able to back out the contaminants gives the most accurate concentration. And this doesn't apply just to DNA, it applies to all nucleic acids being measured. And this is likely one of the largest regions for omic analysis or omic failures in analysis. You look at microarrays that if you've overestimated the amount of DNA that goes into the labeling and fragmentation, is going to lead to poor results. If you overestimate the amount of nucleic acid that goes into a next-gen sequencing library, it'll lead to lower complexity and poorer results. There's also been advances in functional quality control. This is just one slide that actually demonstrates we use genotyping as a function of functional quality control. This is something that's not possible with cubeatless spectroscopy because we're actually looking at genetic information to make sure that we have the right sample. And just really briefly put, for a very low cost, we can look at highly polymorphic SNPs. We have SNPs that are critical for performance. They're representative on commercial microarrays and targeted sequencing panels. It gives you gender, ethnicity, parentage information. It's flexible, it's inexpensive. This combined with Unchained's Analytical QC creates a physical and digital chain of custody that will allow any sample to be used for a downstream application, knowing and being able to predict the quality of that analysis, being able to ensure that the quality of the technology that's being run is spot on because of accurate concentrations and contaminant profile understanding, and then as well as making sure you have the correct sample by creating a unique fingerprint for every sample in this regard. And here's just an example for some sample type specific performance because not all samples are created equal. If you look at DNA from a lymphocyte cell line versus saliva versus whole blood, you can see that analytical and functionally 97% of the samples that pass. But as you look at these other biomaterials, they're going to have a percent pass rate, if you will, of their own because of the overall quality of those samples. So if you're looking at FFPE, the same way we look at quantitation using the Lunatic, the functional quality control is going to be a little bit different because this DNA is all chopped up and degraded for the most part or cross-linked. So the point of this slide is to demonstrate that it's not the overall pass or fail rate of a sample, but you need to understand this from the analytical QC side and the functional QC side to compare within a sample class to know that the overall quality of your samples within a sample class or biomaterial type is reproducible. So hopefully this data and this approach has demonstrated the importance of having integrated quality control and what some of the key technologies are to being able to accomplish that. We must implement technologies that evaluate samples utilized as a part of this omic central dogma. Concentration, purity, contaminant profiles, critical for success for both cell-free and cell-based nucleic acid samples. Adding functional quality control enables us to be able to compare samples in light of different pre-analytical variables. So even though using the Lunatic, we can look at a function of yield, purity, and contaminants, but we also need to see how that's going to impact the performance of a sample. So the integration of analytical and functional quality control is critical. Sample identity and performance prediction as a function of the overall QC program just leads to better science. So if you think about all the concentration and purity measurements that are being made, whether it's for DNA or the purity measurements being made for plasma that we know impact performance, as a biobank, it's our responsibility to create that data, to manage that data, and share it with those that are doing downstream analyses. In the end, the harmonization of these QC metrics supports collaboration on a global level. So these measurements can be made reproducibly across any site, across any lab. In the case of Unchained Lunatic, the consumable, the chip, is easily loaded, and the quality of that read from instrument to instrument, as we have many in the lab, is almost superimposable. So it provides a reference standard for anybody doing quality control. And real-time QC leads to a more fiscally sustainable biobank model or sample collection model, because then you're not storing and managing samples that you know are subpar in quality. And it also allows for a more broad use of these samples because people are assessing the quality of the sample as a function of the whole set they're going to analyze. So really, in short, the Unchained Lunatic provides critical QC analytics utilizing minimal samples at a very low cost. It has become an indispensable part of our QC process within the lab, certainly for plasma, nucleic acid, as we start to develop new applications. I want to thank everyone for their time, for attending the webinar, and I'd be happy to answer any questions, if not immediately following this session, but by email or by text if anybody wants any more detailed information or some collaborative insight. Thank you very much.
Thank you, Kevin and Andy, for that interesting seminar. It's now time for our question and answer session. So to kick us off, I've got a question for you, Kevin, and that is, do I have to read a full 96-well plate at a time, or can I just read a few samples?
So good question. With the Lunatic system, you can just read a few samples, just like you'd think of other 96-well plate systems. So this is the kind of thing that we actually, nowadays we have set up virtual demo options. So we're able to hop on Zoom and kind of show you how to pipette plates and, run your samples even as well. So you really get an understanding of what that consumable looks like and how it all works.
Excellent. Thank you, Kevin. We have another one here for you. And this person is wondering, can I reuse a well?
Okay. So the way the plate works is because it pulls the sample into the reading chamber where the sample is read. There's not a way to clear the sample out afterwards. So each chamber on the plate is one at a time. So once it's read, then that's it.
Great. Thank you. Our next question is, does this instrument come with 21 CFR P11 software?
Yeah, so for 21 CFR Part 11 software, yes, it does. So if you have a GMP or GLP lab and you need to lock down data integrity, then yeah, it's just a software add-on that we make available. Yep.
Excellent. Thank you, Kevin. So unfortunately, that's all we have time for today. So thank you, Andy, and thank you, Kevin, for answering those questions that we did have and for today's informative presentation. And thank you to everyone joining us online. If you do have other questions, please feel free to email me at editor@selectscience.net, and I will follow up with Andy and Kevin to answer those for you. Remember, you can also download your certificate of attendance in the Related Resources tab at the left of your screen. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye, and thank you all again for joining us.